Non-small cell lung cancer mutates continuously. As it spreads, the tumor builds complex defensive grids to block our strongest chemotherapies, causing standard treatments to fail. To break these defenses without waiting a decade for new drugs, scientists rely on drug repurposing, testing approved medications to see if they can effectively target malignant cells. One of the most compelling candidates is ivermectin. Discovered by biologist Satoshi Omura, seen here with a molecular model, this complex compound won a Nobel Prize for its ability to eradicate parasites. Researchers realized the exact biological mechanisms ivermectin uses to paralyze worms might be exactly what is needed to shut down a mutating lung tumor. If you isolate a cancer cell in the pristine, controlled environment of a petri dish, ivermectin executes its job flawlessly. First, it attacks the cell's communication lines. It severs the WNT beta-catenin pathway, stripping the cancer's ability to divide. Then, it shuts down the tumor's transport pumps, locking chemotherapy poison inside so it cannot be expelled. Deprived of its ability to multiply or pump out harmful toxins, the malignant lung cell quickly collapses and breaks apart. If this molecule perfectly destroys lung cancer in the lab, why is it failing to cure human patients in the real world? The answer is that humans are not petri dishes. When a drug enters the living body, it faces the dark, rushing, highly pressurized environment of the human bloodstream. Here, the drug runs into a brutal math problem. Pharmacologists measure the exact dose required to kill a tumor, the IC50, and compare it to the absolute maximum safe concentration a human body can tolerate, known as the Cmax. This chart illustrates the fatal disconnect. The concentration of ivermectin strictly required to kill lung cancer cells requires a massive dose, completely dwarfing the tiny safety limit that human blood can actually hold. The drug's potential is bottlenecked by the rigid mathematical constants of human biology. You might wonder why the drug can't just float through the blood and absorb into the lung tumor. The physical roadblock is a phenomenon called plasma protein binding. Ivermectin is highly lipophilic, meaning it repels water. When these molecules hit the watery environment of the human bloodstream, they defensively clump together. Almost immediately, 93% of the circulating ivermectin is handcuffed by massive, slow-moving plasma proteins. These handcuffed molecules are now physically too bulky to slip through the blood vessel walls and penetrate the dense tissue of a lung tumor. The human body actively traps the drug payload, physically preventing it from reaching the cancer. Online protocols attempt to force a workaround by telling patients to consume massive, unapproved doses to overcome this math. But forcing extreme doses overloads the body's natural safety valves. Specifically, it breaches the highly selective blood-brain barrier. Once inside the central nervous system, the drug attacks the brain's chloride channels. This triggers severe neurotoxicity, inducing deep confusion, tremors, and potentially comatose states. Reaching the lethal dose required to kill the cancer guarantees hitting the lethal dose that destroys the human brain. Patients diagnosed with advanced lung malignancies often face failing salvage therapies and crippling financial costs. They are desperate for accessible alternatives. Algorithm-driven social media and celebrity podcasts take the valid laboratory data and irresponsibly promise human cures, manufacturing a miracle drug myth out of thin air. Because doctors won't prescribe the massive doses needed, patients bypass the medical system entirely, purchasing agricultural veterinary pastes from retail stores. Ingesting non-sterile, bovine-grade compounds packed with heavy metals places an insurmountable burden on the body. Layered on top of standard chemotherapy, it induces rapid, irreversible liver failure. These internet protocols frequently trigger acute toxicity, killing the patient far faster than the lung cancer ever would have. Ivermectin undeniably kills cancer cells, but petri dishes deceive us by completely removing the chaotic human body from the equation. The genuine future of this research is not found in agricultural supply stores. It is happening right now in formal, heavily supervised institutional trials. Scientists are using pharmacological engineering to solve the delivery problem. They are testing lipid nanoparticles designed to sneak the drug past plasma proteins and drop the payload directly into the lung. The National Cancer Institute is also testing ivermectin at safe low doses to see if it can turn tumors hot, making standard immunotherapies far more effective. True medical breakthroughs require safely engineering our way past the stubborn laws of human biology, a reality that cannot be bypassed by desperation and internet hype.